Hello everyone, and welcome to video number 5 of Macroevolution, in which we're going to be looking at something called evolutionary developmental biology, or for short EvoDevo, and we'll throw some fossils in there as well, because why would we not? It's going to be great. So in this material about evolution and paleobiology, we've talked a lot about DNA. We've also talked about morphology, the shape that things are, and those two things are linked by the process of development. So EvoDevo, for short, or evolutionary developmental biology, employs the study of development to investigate evolutionary topics. So it's using the development of organisms, single organisms, to say something about their evolution. Now, there are some words I would like to introduce. The development of an organism is sometimes called its ontogeny. That is the ontogeny of an organism includes its single cell stage from an egg through an embryo all of the way to an adult. So in terms of humans, for example, us growing up from embryo through to grown up, all of that is part of our ontogeny. In organisms that have complete metamorphosis, for example, ontogeny also includes that life stage. And the reason that evolutionary developmental biology is a thing, or EvoDevo exists, is that many trends that we can see in macroevolution can be directly related to development. They often reflect differences in the timing and the duration of different parts of the development of an organism. To choose a really famous example, a giraffe's long neck is due to it extending the period of development of the neck vertebrae, making them grow longer, giving it its long neck. This means that we can study development to provide us with important clues about evolution. For example, modern techniques allow researchers to study when genes are being expressed in a developing embryo using fluorescent molecules. This is a technique that's called in situ hybridization. If you can see where those are expressed in a developing embryo, it helps you understand the physiology of that animal, how it develops in that way. On the right hand side here, you can see some gene expression in a developing Drosophila, so that's a fruit fly embryo. And for example, you can see here these yellow and blue genes are being expressed in a way that suggests that they are responsible for the development of segmentation in this animal. And so that's really, really exciting. For example, by applying this technique across a broad range of organisms has allowed us to recognize in recent decades that some of the genes that control development are shared broadly across the animals. A set of those genes are called the homeobox genes, and those likely have an origin prior to or just at the bottom of the Cambrian period because they're found across all animals that we have studied so far. The same genes are responsible across all of these groups for assigning distinct spatial or positional identities to cells in different regions. So, for example, they allow us to they identify or code for the anterior-posterior axis. On this diagram on the left-hand side here, you can see a series of homeobox genes, and you can see that where they are expressed in a human, developing human, and a fly, shown at the top here, correspond to that anterior to posterior direction in those organisms. That fact allows us to identify which parts of the anatomy of different groups of organisms are homologous to each other, if you remember that word from our phylogeny material. If not, do go and check that out again. And when organisms have very different morphologies, that homology thus allows us to talk about how those groups are related, and the genetics allows us to think about how those distinct morphologies may have evolved and how the underlying genetic mechanisms lead to those resulting morphologies, I should say. Do bear in mind that the genetics of development are mind-bendingly complicated. I couldn't, but there are entire courses on developmental biology out there. And so this is very much just touching on the surface of what developmental biology, or EvoDevo, can tell us. And so understanding all of this when using it to say stuff about evolution is complex. But nevertheless, it has led to great strides. I wanted to give you a single example of that now. In order to do that, I wanted to formally introduce a group that we have touched on elsewhere in this course called the arthropods. So let's have a look at this slide. So I ask, what is an arthropod? Well, arthropods are a group of organisms that have a range of different features of their anatomy in common. They're segmented animals. Their segments, some of them bear ventral jointed limbs. They all grow by shedding their exoskeleton. There's a whole host of different shared characteristics that define the arthropods. The major groups of arthropods that are alive today are shown on this slide. On the left-hand side here, you've got a scorpion that represents the arachnids, and the broader group to which arachnids belong are the chelicerates. Chelicerates are primarily horseshoe crabs plus the arachnids. So that's one living lineage of arthropods. The second here is represented by this crab that represents the crustaceans. Crustaceans are a group that includes crabs and lobsters, shrimp, prawns, woodlice. They're a diverse group with lots of different representatives, many of which are familiar to us today. The third living group of arthropods are shown here. This is a centipede, and this represents the myriapods. That's millipedes and centipedes and their kin. And finally, the fourth major living group of arthropods are the insects, as represented by this butterfly here, but that includes beetles, grasshoppers, all kinds of very familiar creatures. And the group that includes the insects is called the hexapods. So there are some organisms that, say, for example, don't have wings, that aren't quite insects yet, but we call those the hexapods. And these four living groups of arthropods are joined by one extinct group of arthropods, that is the trilobites, which we've talked about elsewhere in this course. So between them, this group makes up the arthropods. This group, I should say, these lineages make up the arthropods. It's also worth noting that crustaceans are actually a paraphyletic group because the insects have evolved from a crustacean ancestor. Insects are just highly derived land-based crustaceans. Why do I include them in a lot of my lectures and why are they important? Well, I think that's well demonstrated by this slide. To a first approximation, every organism that is alive today is an insect. It's very hard to make any reliable estimates of diversity of groups, and so you can take these numbers here as very much indicative, but whichever way you cut it, amongst the animal kingdom, insects make up somewhere between 50 and 85% of described species. So they're an incredibly diverse and successful group. They're also really abundant. They're core to the vast majority of functions in our terrestrial ecosystems. Of the remaining known animals, arachnids make up somewhere between a quarter and 35%. So they're also a really, really diverse group. Between them, the arthropods are this really, really successful group of organisms. Whichever way you cut it, that is true in terms of their species diversity, in terms of their abundance. They're really, really important. And so they're really useful to study the patterns and the processes of evolution, as well as being important to us in lots and lots of other ways. For example, they're vectors for many diseases. Think of mosquitoes, they're insects, they're a member of this group. So the arthropods are a really important group of animals. And they were also a really good example of how EvoDevo can help us understand the nature of evolution and the origin of groups that are around today. So the arthropods are clearly super important. I hope I've made that case, albeit briefly, but relatively forcefully. But a source of long-term argument within this group, which fundamentally impacts on our picture of their evolution, is how the living lineages of arthropods are related to each other. And that colors our picture of how this group has evolved. And a key part of this is the nature of their head appendages. Because arthropods are segmented animals, the first few segments in each of the living groups have been shunted forwards to make up part of the arthropod head. So an arthropod's head is just a series of segments shunted together into a functional unit. And trying to work out which head appendage relates to which between these groups has been the crux of the issues we've faced when trying to resolve their phylogeny and thus trying to understand their evolution. For example, for a long time, people have argued over whether a spider's fangs, the things that it bites with, are what limb they are related to on an insect, shown on the right-hand side here. So the fangs are these these appendages here on this beautiful jumping spider on the left, are those related to the antennae here, or the biting mouth parts, the mandibles of the insect here, or one of these legs, say? This was the subject of discussion for many, many decades, and over the last maybe decade or so, this question has become largely resolved, in part through congruent signals between Hox gene expression domains and the anatomy of these organisms. And that's shown in this diagram here. These relationships have been cemented by scientists looking at the expression of different Hox genes to understand that, in fact, a spider's fangs, the chelicerae here, are homologous to the antennae of a myriapod, and those are homologous to the first antenna of a pancrustacean, of an insect, and of a lobster, say. So that allows us to say with more confidence which segment of the brain of the arthropods each head appendage belongs to, and thus how the different lineages are related to each other. And I can tell you with a far higher degree of confidence now than I would have done even 10 years ago that the myriapods, that's the millipedes and centipedes, are sister group to the insects plus crustaceans, that's the pancrustaceans, and that those two in turn form a clade called the mandibulates that is sister group to this group, the chelicerates, that includes the arachnids. This is a good example of how EvoDevo can help us understand, as my example, evolutionary relationships in a group. But there's another great example within the arthropods of how insects can help us understand a major evolutionary transition, and that is the origin of insect wings. Insects' wings are really, really important. I mentioned already that insects make up somewhere between 50 and 85% of all described animals. They're an incredibly successful and diverse group, and in part that's probably because they can fly. And EvoDevo can help us understand where these wings have come from. Insects came onto land at some point before 315 million years ago. Exactly when is quite a big question. If you want to learn more about that, you can see more in my videos about terrestrialization. But when insects came onto land, there was or I should say the group that includes insects, so hexapods came onto land, and then insects evolved flight at some point. There was probably an adaptive radiation into this huge number of different insect groups that are alive today. But insect wings are really interesting because they differ fundamentally from those in other flying groups of organisms. If you think about birds, bats, or pterosaurs, all of those do's and dudettes have modified a forelimb like our arm, sorry, let me get that right, like our arm, this thing here, into a structure that will allow them to fly. That is not the case in insects. If you look at all of the winged insects in this beautiful image here, you can see that these creatures with wings still have legs on each associated segment. So the insects have not co-opted a limb into being a wing, as has happened in the other groups of organisms that fly. So that means that asking where insect wings come from is actually really, really challenging. And I've got to tell you, sadly, the fossil record doesn't answer the question either. There's a big gap in the fossil record of insects up to the appearance of the first winged insects 350 million years ago, in which these guys and girls evolved their wings. So we don't really capture it in any kind of complete way in the fossil record. And so there's this really big question here about where insect wings have come from. And we've been able to answer that in the last 5 or 6 years, thanks to the development of evolutionary developmental biology models to allow us to ask that particular question. This slide is based on a few papers that take EvoDevo and use it to try and answer these questions. These papers compare the development of an insect, shown at the bottom left here, to a crustacean, shown at the top left. The details of what they've managed to reveal are really wonderful. I would encourage you, if you have time and you're interested in this, to check out those papers. Or if you want a more gentle introduction, the introduction by Smith and Jokush that I've put on the slide here to the other two papers is a really nice, accessible way of getting to this. So please do check those out if you're interested. But in brief, both of the authors of these papers from 2020, Bruce and Patel in 2020, and Clark Hatchell and Tomoyasu, in 2020, look at the development of either the limbs or the body in these two organisms. And both authors in both papers agree that the lateral body wall of insects is homologous to the proximal most leg segment of crustaceans. So, in both of these papers, the authors identify that a segment of the leg of an ancestor has been incorporated ultimately into the body wall of an ancestral and wingless member of the group that includes the insects. They also agree, both of these papers, that the wing incorporates body wall components that are derived from crustacean legs. So in both of these papers, we've got this picture of a multi-segmented limb and a segment being becoming part of the body wall, and eventually that developing into a wing, which is a huge step forward from what we thought about the origin of wings before this point. As shown on the right-hand side here, they differ in the details. They disagree over precisely which element then went on to evolve wings. They essentially both ask, is it just the dorsal portion of the body wall, a piece of the anatomy we call the tergum, or is it the tergum and another region, a more lateral part of the body wall called the pleuron, that went on to form the wings? And they differ in what they think ultimately went on to evolve to become a wing. A more recent paper by Ode, Maiter, and Nimi in 2022 supports the first hypothesis, actually, that the tergum, this dorsal plate, is the core element that evolves into the wings. And so that's really, we're starting to build, I think, a consensus about the origin of insect wings and the sequence of events that went from a crustacean to a flying insect. And I just wanted to highlight also that Odie and Prokop in 2025 support this first hypothesis and look at Paleozoic fossils and identified both thoracic and abdominal lateral body outgrowths, which they suggest may be transitional wing precursors, and suggested that they possibly had a role as respiratory organs in aquatic or semi-aquatic environments. So fossils are starting to fill in a few of the gaps in this sequence as well. It's a really complex but really cool picture that we're starting to build up in recent years. So overall, as a conclusion to this slide, I would say now that we can say with confidence that insect wings have evolved from existing limb structures via an intermediate transition from a proximal leg segment into the body wall and then into wings, which is a really cool thing to be able to say about the insects. So I hope you found that interesting. I wanted to finish by talking about Paleo-EvoDevo. So you may be sitting there being like, okay, Russell, you've talked about all this stuff, but what's that got to do with paleontology? It's a evolution and paleobiology lecture. Where's the paleo bit? And you may be saying, but you can't get embryos from the fossil record, right? And I would answer to that, surprisingly, wrong, actually. The preservation of embryos in the fossil record isn't unheard of. And there are a few really famous examples. The fossils that are shown on the left-hand side here are fossils that were discovered in a phosphate mine in China. This is quite a famous fossil deposit known as the Doshan2 microbiota, or I should say the fauna, the biota from this is known as the Doshan2 microbiota. The dates from between 551